rock and roll. Okay, yes, ma'am. When we take the final? I haven't seen the final schedule, but I think they will probably all be on on Thursday the 15th would be my guess. Yeah, I hadn't seen anything, though. I have not seen anything about that. Pardon? Like the 12th? Yeah, again, I haven't seen the final schedule. I don't really know what they're going to do about that. You know, it's the first time we're doing the 8 weeks, so well, look at you. Oh, man. Oh, this is cool. I know. Thank you, Monica. That's very cool. Um, yeah. Her mother's partner has a nursery, and so they're donating this, which is very, very nice, because that will be a good plant. Absolutely. Okay, there are just a couple of things that we didn't actually get to yesterday, and we need to finish out that chapter 6, and it's not much. There's a couple of terms that I didn't give you, and I feel like I need to give you because it's just two important terms. One is called a substrate level. Now these terms you're going to go, oh my God, I got to remember that. I understand. Okay, substrate level phosphorylation. You're going, okay, I've just died to hell. I am in hell now. I've got to remember that term, substrate level. At one point, we did talk about the fact that when I was younger, like last year, no, I don't know. I don't remember how old I was. But my parents gave me a chemistry set for Christmas, and I just loved it. I had no idea what I was doing. But I mixed two chemicals together, and I was holding that test tube, and it got so hot I had to put it down. Okay, that's what you call an exergonic reaction. Some reactions just spontaneously give off energy. So I don't remember what molecule A was, nor do I remember what molecule B was, but obviously I produced molecule C. Something happened. So I produced something else from those two things, but it also released energy. In this particular example, with what I was doing, it released heat. But you know, there's all kinds of energy forms out there, from magnetic energy to light energy to blah blah blah, kinetic energy. You know, we'd go on with that. But what the cell wants to do with reactions that spontaneously release energy, which we call, this is an exergonic, doesn't consume energy. You know, most reactions that happen, we tend to think they require energy. You got to put energy into it to make it work. Well, some of them you don't. It just spontaneously happens. Put this together with that, and it just spontaneously occurs. You don't have to heat it up. You don't have to nothing, and it releases energy. Okay, so that's exergonic. If you had to put energy into it, it's exergonic. We've been through that, although this semester is a big blur for all of us. It's like, what? We did what? We covered biology? Really? Was that a whole semester? 8 weeks? Really? Wow. That was crazy. Whoa. Seriously. Anyway, so what a cell wants to do when this reaction, something like this happens, and energy is just spontaneously released from that reaction. It's like, you know, we could let all that energy go out and just let it go off as heat. You could, but the cell is not that stupid. The cell is like, okay, I can do something with this energy. So what it will do is it will take ADP, if it's enough energy, ADP, adenosine diphosphate, and just a PO4, a phosphate, inorganic phosphate, PI, and it puts the third phosphate onto the other two phosphates. So what I'm generating here is ATP. So there were a couple of places, like in glycolysis, where we actually generated some energy. It took 2 ATPs to get it all started, but when you were finished with it, there were 4 places where we generated the ATP. So we actually made 4 ATPs. But of the 4, you would use 2 to redo that, to get that whole thing started again. So really, you got a net gain of 2 ATPs. Okay, those 2 ATPs are made exactly like what I just described. It's a substrate level phosphorylation, meaning that you have a reaction and it just automatically gets off some energy. It's like, you know, me and my chemistry set. Automatically energy came out of it. And if that happens in the cell is smart enough to go, okay, wait a minute, you know, I'm just not going to let all that go off as heat. That would really be stupid. I've got energy here. Let's do something with it. So we put a phosphate on ADP. So you can generate a little bit of ATP, not much, but 5% of the ATP, 5%, is actually produced this way, exactly, by doing this. The other 95% of the ATP we manufacture is in the mitochondria, which we described yesterday. Take the hydrogen, pull it apart, let them slam back together, get enough energy to make ATP, and we did that. So, but this particular method, again, only produces 5% of your ATPs, but, I mean, 5% is better than no percent. You get a little bit of energy out of it, so that's cool. That's good, 5%. So that is a net gain of 2 ATPs, and this is how it is produced, substrate level phosphorylation. And it is a phosphorylation, and we've talked about this before. A phosphorylation would be just to pick up a phosphate group. So have you ever been phosphorylated? You've been phosphorylated? Never. See, I could tell she's a virgin there. So if this is a phosphate group and I let her pick that up, she's phosphorylated. So the virginity is totally destroyed. Yeah, she is phosphorylated. She picked up that phosphate group. That's all a phosphorylation means. It just means to put a phosphate onto something. You can put it on a sugar. You can put it on whatever. But if you put it on ADP, you've made ATP. So it takes some energy to do that. Of course, adenosine diphosphate, you have 2 phosphates. So that thing is going to be negative because you've got 4 oxygens around each phosphate group. This piece right here, the PI, has 4 oxygens around it. So it's negative also. So to force that PI and make it attach to the ADP, they're both negative. You don't want to just automatically attach. You got to have energy to make that happen. So this energy comes right out of that exergonic reaction. You have a reaction just spontaneously releasing energy. So the cell goes, I think I'll just do something with that. And it uses that energy to put a phosphate on ADP. So you generate a little bit of ATP, adenosine triphosphate. Like I said, that's a 5% of our energy production, not a whole bunch. So it is a phosphorylation. Substrate level means that the energy that is released, this energy right here, that came out of that, it didn't come out of C. C doesn't have it. It must have come from A and B. Must have. It didn't come out of C. C's a product. So that energy came out of A and B. Well, what are those? Those are substrates. It's what you begin the reaction with. So I have 2 substrates, A and B. The energy came from somewhere in here. It's actually bond energy, but it came out of this, and it was used to put a phosphate on the ADP. So once you did that, you obviously made your ATP. And that's all a substrate level phosphorylation is. It sounds really complicated. You know, you pitch that term out to you know, at the dinner table. It's like, well, you know, what'd you learn in biology today? Well, we talked about substrate level phosphorylation. You know, they're going to be all impressed and go, wow, you know, it's like, yeah, but if you really knew what that was, you wouldn't be too impressed. You got a reaction that releases energy. Use it to put a phosphate on ADP. And you now have stored it. You stored it, ATP. You can store it. just hangs out in that cell until you need it. And then you pull that phosphate off, and it releases that energy. So it's just a way to store energy is what it is. Anyway, substrate level phosphorylation. That's as complicated as that gets. You know, people, the whole phrase substrate level phosphorylation, it sounds really complicated, but you look at it and you go, no, not really. The energy came out of the substrates and you used it to put a phosphate on ADP. End of story. Well, not very complicated. Okay, the other thing that we did is we kept taking all these hydrogens off of really sugar because that's what we started. We put them on carriers like NAD or FAD. We just threw some hydrogens on carriers. And then we took them into the mitochondrion. Some of them were actually produced in the mitochondrion. And then we started taking those hydrogens and we started pulling them apart. So hydrogen, of course, you just have two particles. You have a proton and an electron. Protons are positive, electrons are negative. So we take the hydrogen and we pull it apart. And then we allow a really strong positive charge to build up. Let's just draw a mitochondrion up here. I like visual images. So it is the male member of the human race that tends to watch more porn than it is the female. More pornography. It's like, you know, it's like they say 80% of all web traffic is pornography. 80%? Like, really? Yeah, that much. It's a lot. 80%. Like, whoa. You know, I wished I cared enough to even pull it up. You know, it's like, I don't even care. You know, if I were 21, I probably would. You know, but at 58, it's like, I don't even care. I don't care what they're doing. It make me any difference. It's not interesting at all. You know, I'd rather find other things to do. Rearrange the living room. You know, hey, it's more fun. I know. You are a sad case, bro. Oh well, reality bites. Okay, so we took all these hydrogens and we put them on carriers like NAD or FAD, and we ran them through this series of where we take those hydrogens, NADH2. So we put some hydrogens on that, or we put them on FAD, FADH2. And so we took those hydrogens and we took them to a carrier, and those carriers do have names. Those things are referred to as cytochromes. Cytochrome, and they are enzymes. They're proteins. Okay, cytochrome enzymes. They have names, and they all, each one has a specific name. But I'm not getting into that. We're not going to bore you that much today, hopefully, although I can be quite boring. I usually have to make a pretty strong effort at it. I don't find myself boring at all. You may. I don't. Okay, so we're just moving hydrogens from one carrier cytochrome to another carrier, just pulling two hydrogens and then moving them from here to here, pulling those two hydrogens and then moving them from here to here. All right, two hydrogens. And then at some point, because we have oxygen in here, which is electronegative, it has a negative charge. The reason it has a negative charge is because I take my hydrogen, which really you just have two pieces in hydrogen. You have the one proton and you have the one electron. That's it. It's not any more complicated than that. And you've heard me say this, but I think it's absolutely the most beautiful system in the world, where you can take the simplest element on the earth, the simplest element, and you can use that to power every living creature on the planet. And it is the most efficient energy system on the planet. Most efficient. You just pull the two particles apart, the positive, the negative, and you build up a really strong, because the electron's going to go here. So electron goes in here. The proton goes out here. protons out here. And you keep doing this with all those hydrogens. So we had several places where we put hydrogens on carriers. We got them off sugar mostly. Okay. So we build up a whole bunch of positive protons out here, and you build up a whole bunch of negative oxygens in here since it is electronegative. And then you have this enzyme, and that enzyme has a name. It's called ATP synthase. It ends in an ASE. That tells me it's an enzyme. And then you pull the positive in, and you pull the negative in, the oxygen. They tend to pull in. Certainly they would attract. One's positive, one's negative. So they'll attract and slam together. And when they do slam together, if I get my electron, I pulled them apart, but if I get my electron back together with my proton, put them back together, which is what we're doing, once I do that, I've created hydrogen. So the hydrogen's on the oxygen, so what comes out of this thing is going to be water, totally harmless. That's what they call metabolic water. You've ever heard that term? Metabolic water. I mean, if you stay in the field of biology, go into pharmacy, go into whatever, you're going to hear metabolic water. What they're talking about. At the very end of cell respiration, you make some water. It's metabolic water, not anything you actually consume. So you've rebuilt your hydrogens, you pull them apart, and then you rebuild them. And this time they're on oxygen. The other thing that comes out of that, of course, is energy. So you get enough energy to make ATP. So that energy is used to take ADP plus inorganic phosphate, PI, and you generate ATP. So this is how we make 95% of our ATP. So most of it is made by doing this. Taking hydrogen, pull it apart, slam it back together, you get enough energy to make ATP. To put a phosphate on an ADP and you've generated ATP. This system by which we do this, which I look at that and there couldn't be anything more simple than that. Take hydrogen, pull the positive and the negative particles apart, build up a really strong positive between the inner and outer membrane, a really strong negative charge, and at some point they're going to pull together and release energy. You make water, which is harmless. That's your metabolic water, and you release enough energy to put a phosphate on ADP. So we generated some ATP, which is the stored energy. You can store it. But if you want to walk across the room or whatever, think, it's going to take some energy to do all of that. So then we begin to use it. And when you're totally out, well, you know, you just better stop. You don't even have a choice. It's called fatigue. You stop. Can't go any further. And you can fatigue your brain if you study too much. You know, if you're one of those people that like to study hour after hour after hour, you know, it's like you can really concentrate real hard on that, you're doing it wrong. You know, what you need to do is read through it. Or actually, I like to write it from beginning to end, all my notes. And then, you know, when I'm finished with that, take a break. Take a 30, 45-hour break. Go do something else. Come back to it, rewrite it again. Take a break. Come back, rewrite it again. Take a break. You know, you don't want to fatigue the brain because then nothing's going in. And you get frustrated and upset, and I always did. It's like, well, this isn't working. I'm not understanding this. Well, you're fatigued. You just don't do that. It doesn't work. So everybody needs rest. I mean, that's just part of life, and you have to do it. And if you're not, you're not going to be successful. You have to. Anyway, this host system by which we do this, it has a name. It's called chemiosmotic coupling. You're going, oh, another nasty-ass term I got to put in my head. Thank you very much. I didn't come up with it, but if you want to shoot the messenger, okay. I don't mind. Doesn't hurt. Yeah, the whole process. process. You pull the hydrogen and slam them back together to get enough energy to make ATP. Called chemiosmotic coupling. Many times I don't like to just immediately give you the nasty term where you go, oh my gosh, this is going to be ugly, and you just dread it. You know? Show the student how it works, and they're going, okay, that's how this thing's going to work. And then throw that nasty-ass term on the end of it. They're like, oh, okay. Substrate level phosphorylation. Oh, okay. Like, everybody's surprised. Well, that wasn't too bad. Beats the heck out of putting in a carburetor. As if I knew how to put in a carburetor. I have no clue. I couldn't even identify a carburetor in the car. It's like, it's in here somewhere. Carburetor. Goodness. Who named that? Let's figure that out. Who did this to us? Carburetor? Are you kidding? What does that even mean? Is that a term? Really? Okay, I'm sorry. I did take my meds this morning, though. You'll be proud of me. It would be worse if I hadn't. Okay, so that's about all I want to do with Chapter 6. So anyway, and you've got, of course, your test, which some of you may have taken yesterday, and some of you may take today. And, of course, the module test, I guess it's still up. Can anybody tell me, because I really hadn't looked? It's not up anymore? Module 2? It's not? I can reopen it. Yeah. I'll reopen it for today if you didn't get to it. Anybody not get to it? Just so that you know, at the very end of the semester, I reopen everything. So if you didn't get to an assignment, it's like I didn't have time to do that one, I reopen everything. It's like I can now do it. So keep that in mind. It's like, I didn't have time. I had things that had to happen, and I couldn't do it. Don't worry about it. I will open it for about a week. Not just the last week of the semester, but I will keep everything open for about a week if you need anything that you need to go back and revisit and work on. So you have time to finish stuff for sure. I don't know. I think the more you can help a student, I mean, what? That's what we're here to do, right? I think that's the whole job. The job is to help the student get through this stuff. That is the job. I just happen to like to have fun. Okay, so we're going to start Chapter 7 today, which is on photosynthesis. So you kind of have to just take your brain and split it in half, because you've got cell respiration and alcoholic fermentation or ethanol fermentation, and then you've got lactic acid fermentation, you've got glycolysis. You got all of those reactions going on that somehow you have to file away in your head. And then we do photosynthesis, which is also a whole bunch of reactions. And but you just keep them separate, because that Chapter 7 is is not even tied to the others in terms of testing, nothing. I get all that done, and then I do photosynthesis, and you get tests just over that one chapter. And I find that to be very successful. A lot of people don't. They throw it all in together, and you can test it over all of it, which I think just really messes up the head quite a bit. So I keep them separate. So anyway, you have all that other stuff. We'll just kind of walk right past that, and then we'll get into photosynthesis, Chapter 7. So that's really what we're trying to do today. Get a couple of terms away on cell respiration. So let's do photosynthesis. Synthesis means to produce, to create. Photo means we're going to use light to make something. What we're making is sugar. And plants have the ability to do that. They have the ability to take sunlight And then they also take in some water, typically out of the ground, unless it's a water plant and they're surrounded by it, so they just get it right there. And carbon dioxide, typically get that out of the air. So let's say that it's a land plant, not a water plant, but land plant. You've got this leaf, all right, and on the lower surface, you have these things that kind of look like this. They look like lips. And they're called stomata. Stomata. As long as they're open, we can get oxygen out because a plant will produce an excess of oxygen. They need oxygen for cell respiration because they do exactly what we do. They do glycolysis, and then they do the preparatory reaction, and then they do the Krebs cycle, citric acid, and then they do the electron transport. And they generate a lot of ATP. That way they can grow. Important. They start out like a little seed, and then they grow, and next thing you know, you've got an oak tree, whatever. So yeah. So, but the other thing that has to happen is they have to pull in carbon dioxide out of the environment, CO2, because that will be building material in order to produce sugar. Sugar, of course, is C6H12O6. So, you know, this is our sugar we got to manufacture. Well, the carbons and the oxygens, you know, out of CO2. We also need water in order to do that. So the way they usually put this is they put on photosynthesis, they put a 6 CO2 plus 6 waters. Actually, I think it's maybe 12 plus water, and then using the sunlight. That's what I thought, 12. Thank you. All right, so try to balance this thing out, although I'm never really too concerned about balancing an equation. You just know what you put into it and what you get out of it. And what you get out of it is sugar. Plus, you make some oxygen. And the plants use that, so some of it goes into the mitochondrion. There's an N at the end of that. For cell respiration. The oxygen that goes into the mitochondrion is used to pull the electron off the hydrogen. Then you throw the proton out between the outer and inner membrane, and the oxygen pulls the electron, so you build up a really strong positive and a really strong negative. Eventually they slam together and you make ATP. We did that. It's called cell respiration. They do that too because they need to have ATP in order to grow. So they make their own sugar. You know, we consume that either as sugar, or we consume protein and we convert some of it into sugar, or we consume fats and we convert some of that into sugar. You know, plants make it. And they make their own oxygen too. They make their own food and they make their own oxygen. I'm thinking, this is a pretty sweet deal. They just open up some holes called stomata. Open up these holes right here and take in some CO2. They make their own sugar and they make their own oxygen. They're pretty well set. They do the whole cell respiration thing, exactly like you and I do it. But they can make their own sugar and their own oxygen. So some of that oxygen they do use for cell respiration, but some of it will go out. So this goes to the environment, which you and I breathe, and your cat gets to breathe it, and your dog gets to breathe it, and the earthworm gets to breathe it, and the snail, and all the animals get their oxygen. You know, and back when, there really wasn't any free oxygen. Free oxygen didn't begin to accumulate in the environment until certainly we had plants evolve, or at least algae evolved, which are photosynthetic, which are not plants. Algae are not plants. They're not advanced enough to be a plant. To be a plant, you have to have a certain amount of internal anatomy. The internal structure has to be at a certain level before you are technically a plant in the plant kingdom. Algae are not that complicated, so they're not in the plant kingdom. They're in a category which would be the protists, like an amoeba, paramecium, blah, blah, so forth, but they're not plants. But they are photosynthetic. And then from that, eventually you did evolve plants. So the oxygen goes off. It's just a waste product. I mean, they use it in the mitochondrion for cell respiration, but also they produce an abundance. Thank goodness. That's great. You know, and people talk about, you know, well, okay, we burn fossil fuels and we produce a lot of carbon dioxide and the CO2. Well, that causes global warming. Guess who loves that? Plants. They're like, man, I got a lot of CO2. I can make a lot more sugar. I can grow even faster. Plants are not, no, they're not going, oh, this is so horrible. Their automobiles are shooting out all this carbon dioxide. They're going, that is so cool. You know, and typically that is what causes eventually the cooling of the environment. You know, you go through global warming and then you go through the global cooling is the second thing that comes along. Now, the time frames, of course, are typically very long. In this particular example, with what we've got going on today, that time frame will be pretty short because we're actually having global warming happening at an exponential rate. It's happening very quickly. We're warming the environment because the carbon dioxide in the atmosphere forms like a little blanket, and the sunlight can get through the blanket. And I guess probably a lot of you understand this already, but I don't want to tell you things that you already know. But if this is a CO2 layer, the thicker that is, has no effect on the amount of sunlight that can get in. So sunlight can actually get through the CO2 layer. It's a very short energy form, so light can get in, sunlight. But then heat is a very long wavelength. All right, and the heat cannot get out. So it gets stopped. This is heat. So the more CO2 we have in the environment, the thicker the blanket is. It doesn't affect the amount of light that can come in. It just keeps coming in. But then it's converted into heat. Go outside and pick up a rock after it's been out in the hot sun for a period of time. It's going to be hot. But you know what? It's converted. The sunlight is absorbed in there, and it converts from sunlight. Obviously, the rock isn't going to glow, so it's converted from light into heat, and the heat wavelength is like this. You've seen it. Hood of a car, you know, real hot, and you can see this like that. Well, of course, it just traps it. So, yeah, global warming happens. But the more carbon dioxide we have, the faster plants will grow, because, you know, that's what they need in order to produce their sugar. So they're just happy as clams. You know, they're just like, this is good. Yeah, why don't you drive a big old truck? Yeah. Keep those things moving, because I'm loving it. But once they start absorbing all that CO2, the layer of carbon dioxide, let's say your layer is really thick, like that, so you got a whole bunch of CO2. Once they start absorbing that, your layer gets thinner and thinner and thinner, okay? And as the plants continue to grow and thrive on that carbon dioxide and produce more sugar and grow faster, so their growth rate increases, and they're taking in that CO2 out of the air. So at some point, your carbon dioxide level gets very, very, very thin. And what that means then, at that point, because the plants absorb the CO2, and this has happened throughout time, when they absorb the CO2, then this layer right here gets very thin, then the heat can, at this point, actually escape into outer space. So it actually leaves the planet. What do we have? We have global cooling. You end up with an ice age. Sure. You know, 3/4 of America 10,000 years ago used to be covered with an ice sheet. That wasn't all that long ago. You know, and now we're having global warming, but it's happening really, really fast. So the plants are going, ooh, it's great. I'm getting really huge. And then they're going to suck out all that carbon dioxide layer, and then you don't hold the heat in anymore. And then all of a sudden, all the heat goes out, and then you get global cooling, which then you end up with an ice age, which will happen like crazy fast this time. It'll really happen real fast. So you keep having these cycles of warming and cooling. And people talk about that, you start talking about global warming, they go, well, it's always done that. You go, yeah, I know, but really not at this rate. This rate is going to be crazy. Your great-grandchildren may experience the ice age. It's like all of a sudden it gets freaking cold, and that'll be real interesting. I don't know how hot it'll get. I don't know what all that's going to do, but it's going to have a major impact on what survives and what doesn't survive. And then it's going to be thrown into an ice age, and what survives and what doesn't survive. It's going to be kind of a crazy thing because it happens so fast. And the problem with that, if it happens slowly, things can adapt and evolve. But if it happens so quickly, you don't have enough time for evolution. You have to change the gene structure in order to get the evolution to occur. If it happens so fast, you don't have enough time to change the gene structure. That takes time to do it. So I don't know what will eventually happen with it. We may just wipe out every living thing on the planet. None of it can survive. Real hot, and then bam, just freezing cold. And there's no time for evolution. So bye bye, bye bye, bye bye, bye. It's like nothing survives. I tend to be a little bit more optimistic about that. I think some things will survive, and then those will continue to evolve. So I don't think all life will be wiped out. I think a whole bunch of it will be, for sure. And again, it's not going to happen in your lifetime. I certainly hope not in your lifetime. But, hopefully most of you will have children, and they will have children, and they will have children. So, at some point, somebody's going to be affected, for sure. And we're affected to some degree, but not on the level of what I'm talking about. Okay, anyway. So, drive that big old dually. Fill that puppy up, Everybody go, wow, it's a dually. I'm like, oh, that just really sucks. Bad. Yeah, bad. Not a good thing. All right, so photosynthesis. Oh, I didn't throw in, we also make some water here too. Metabolic water. I think it's 6. Again, I'm not ever too concerned about balancing all that out, but okay. So, and let's talk some more about stoma or stomata. The whole itself has a name. The entire structure, which really consists of 2 cells. So if I were to take one of those and enlarge it, let's just say we take this thing and make it bigger. Okay. Really what you've got is kind of like a couple of bananas. One goes this way, like that, and one goes this way, sort of like that. 2 bananas. Okay, these are called guard cells. That's a guard cell. This is another guard cell. And the whole unit is what we call stomata. The whole section. That's a stomata. All right, the whole thing. The whole itself is called a stoma. That's Latin for whole. If you don't like someone, you can just call them a stoma. You're such a hole. A hole on earth, you're a stoma. That's pretty rude. They would know what you're saying, but what a stoma. What a freaking hole. Did you really have to occupy this space? Was that necessary? Gosh, think about this. Do some things that are good. Don't be a stoma. Let's make the world a better place. No one wants to be a stoma. Big old hole. All right, enough about those stoma. All right, so let's talk about photosynthesis. We don't have much time, but there are two major reactions that occur on photosynthesis. That's a Y. Photosynthesis. Two primary or major reactions. All right. One of these is called the light-dependent reaction. Obviously, you need light. Why else would they call it light-dependent reaction? Which, if they need light, that's a P, dependent reaction. If you need light, you need something to absorb the light, so that's where the pigments come in. You know, like in a plant, the primary pigment you get there's a chlorophyll that's green. You got to have chlorophyll in there. Of course, there are all kinds of different wavelengths that we get from that. So this is where the chlorophyll is used. Need something to pick up that light, so we need pigments. Chlorophyll mostly. And the other one, you don't need the light at all, which means it can happen at night, it can happen at light has no impact on this set of reactions whatsoever. It's what they call the light. They call it the dark reaction. The dark reaction, well, that kind of makes most people, and that's what I learned, the dark reaction. I didn't learn light independent, but that always makes you think, okay, well, one of them happens in the daytime, one of them happens at night. Light independent, which doesn't require light, happens whenever. Middle of the day. You just don't need light for it. Okay, it's light independent. Anyway, so that would be our second major reaction, light independent reaction. All right, so let's talk about the light dependent reaction first. And we can divide that up also. So the light dependent and the light independent, you don't need light for that part at all. So we don't need pigments. We're not absorbing light. Wouldn't be necessary. So the light dependent reaction, you have two forms, two types. Say two types, two forms. Call it whichever. Anyway, one type is called the electron. I just abbreviate that. Oh, I'm sorry. Cyclic let me think. Books do it differently. We learn cyclic photophosphorylation, which means you're picking up a phosphate. Your book uses light or cyclic. I had to write it down because cyclic electron pathway, that's what they use. Cyclic electron pathway. Again, different books, different terms. Cyclic electron pathway. All right. And the cells that do this, so, regardless of what the reaction is, pretty much always learn what's the name of the process, what cells do it, where does it happen, what do you those are the things. It's always those things. Name of the process, what do it, where does it happen, what do you start with, what do you produce. After you've seen so many different reaction series, like at some you get to take